The work of Gregor Mendel. What would you like to inherit? Where does an organism get its unique characteristics? An individual's characteristics are determined by factors that are passed from one parental generation to the next. Every living thing, plant or animal, microbe or human, has a set of characteristics inherited from its parents. Asexual reproduction, or parents for sexual reproduction. The delivery of a characteristic from a parent to offspring is called heredity. For asexual reproduction, the child is identical to the parent. The scientific study of heredity, known as genetics, is the key to understanding what makes each organism unique. The modern science of genetics was founded by an Austrian monk named Gregor Mendel. Mendel was in charge of the monastery garden where he was able to do experiments that changed biology forever. Mendel carried out his work with ordinary garden peas, partly because peas are small and easy to grow. A single pea plant can produce hundreds of offspring. Today, we call peas a model system. Scientists use model systems because they are convenient to study and may tell us how other organisms, including humans, actually function. By using peas, Mendel was able to carry out, in just 1 or 2 growing seasons, experiments that would have been impossible to do with humans and that would have taken decades, if not centuries, to do with other large animals. Mendel knew that the male part of each flower makes pollen, which contains sperm, the plant's male reproductive cells. Similarly, Mendel knew that the female portion of each flower produces reproductive cells called eggs, male parts and the female parts on the same pea flower. During sexual reproduction, male and female reproductive cells join in a process known as fertilization to produce a new cell. In peas, this new cell develops into a tiny embryo encased within a seed. Pea flowers are normally self pollinating, which means that sperm cells fertilize egg cells from within the same flower. A plant grown from a seed produced by self pollination inherits all of its characteristics from the single plant that bore it. In effect, it has a single parent. Mendel's garden has several stalks of pea plants that were true breeding, meaning that they were self pollinating and would produce offspring with identical traits to themselves. In other words, the traits of each successive generation would be the same. A trait is a specific characteristics of an individual, such as seed color or plant height, and may vary from one individual to another. Mendel decided to cross his stalks of true breeding plants. He caused one plant to reproduce with another plant. So he took the pollen from one and fertilized the other plant. To do this, he had to prevent self pollination. He did so by cutting away the pollen bearing male parts of a flower and then dusting the pollen from a different plant onto the female part of that flower, as shown in the figure. He had removed the male portion of this plant, this flower, and dusted the pollen onto the female portion. Cross pollination, taking one plant to another, not self pollination, which would be the same plant to the same plant. This process, known as cross pollination, produces a plant that has 2 different parents. Cross pollination allowed Mendel to breed plants with traits different from those of their parents and then study the results. Mendel studied 7 different traits of pea plants, each of which has 2 contrasting characteristics, such as green seed color or yellow seed color. Mendel crossed plants with each of the 7 contrasting characteristics and then studied their offspring. The offspring of crosses between parents with different traits are called hybrids. When doing genetic crosses, we call the original pair of plants the pea. or parental generation, the P generation, the parent. And these are the categories, and these were the two options he saw as describing these. Their offspring are called the F1 or first filial generation. For each trait studied in Mendel's experiments, all the offspring had characteristics of only one of their parents, as shown in the table. So the F1, the first filial, had round, yellow, gray. Notice that these are just one of the two options from the parents. Chose only one of these two. In each cross, the nature of the other parent, which did not show up, it seemed to have disappeared with its trait. From these results, Mendel drew two conclusions. His first conclusion formed the basis of our current understanding of inheritance. An individual's characteristics are determined by factors that are passed from one parent generation to the next. Scientists call the factors that are passed from one parent to offspring, scientists call these factors genes, the things that pass from one parent to the offspring. Each of the traits Mendel studied was controlled by one gene that occurred in two contrasting varieties. These gene variations produced different expressions or forms of each trait. The different forms of a gene are called alleles. Mendel's second conclusion is called the principle of dominance. This principle states that some alleles are dominant and others are recessive. An organism with at least one dominant allele for a particular form of a trait will exhibit that form of the trait. An organism with a recessive allele for a particular form of a trait will exhibit that form only when the dominant allele for that trait is not present. In Mendel's experiment, the allele for tall plants was dominant and the allele for short plants was recessive. So plant height, tall, and a short will breed together, you're going to get a dominant characteristic, which was tall in the F1, the first filial generation. Likewise, the allele for yellow seed was dominant over the recessive allele for green seeds. Yellow crossed with a green, you'll get a yellow as dominant in the first F1 generation. Can you choose your own inheritance? During gamete formation, the alleles for each gene segregate from each other so that each gamete carries only one allele for each gene. In actuality, alleles and inheritance are more complex, but to gain understanding, we will use an example simply with two alleles, one dominant and the other recessive. Mendel wanted to find out what had happened to the recessive allele. To find out, Mendel allowed all 7 kinds of F1 hybrids to self-pollinate, and the offspring of the F1 crosses are called F2 generation, the second. The F2 offspring in Mendel's experiment are shown. So a tall to the short produced 2 F1 generation, a tall and tall, of course, expected that dominant trait. But then the F2 produced a tall, and a short, which was not appearing in the F1 generation. When Mendel compared the F2 plants, he discovered the traits controlled by the recessive alleles reappeared in the second generation. Roughly 1/4 of the F2 plants showed the trait controlled by the recessive allele. Mendel assumed that a dominant allele had masked the corresponding recessive allele in the F1 generation. There's a recessive gene in both of the F1 plants, but it's masked. The reappearance of the recessive trait in the F2 generation indicated that at some point the allele for shortness had separated from the allele for tallness. How did this separation or segregation of the alleles occur? Mendel suggested that the alleles for tallness and shortness in the F1 plants must have segregated from each other during the formation of the sex cells, or gametes. Let's assume that each F1 plant all of which were tall, inherited an allele for tallness from its tall parent and an allele for shortness from its short parent. So it has a capital T for tall and a small t for short. So here are the gametes. The F1 generation had one from the tall and one from the short, and here we list them. Tall gamete, short gamete, tall gamete, short gamete. When they're combined, this one and this one, choosing one of each, would be tall. This one and this one combined together, they would be also tall because tallness is dominant. And this one also, combining the small t and the large t, would also be a tall plant. But finally, the last one, inheriting the short gamete and the short gamete from each of the F1 generation, would inherit a small t, and that would be a short. Homozygous means that they're both T and T the same gamete. Heterozygous means you have one of each. And then homozygous, also the same, in this case, too short. When each parent, or F1 adult, produces gametes, the alleles for each gene segregate from one another so that each gamete carries only one allele for each gene. They'll separate into tall gamete and short gamete. A capital letter represents a dominant allele. A lowercase letter represents a recessive allele. Dominant, recessive. Each F1 plant in Mendel's cross produced two kinds of gametes, those with allele for tallness, capital T, and those with allele for shortness, small t. Whenever each of the two gametes carried the T allele, small t allele, and then paired with the other gamete to produce an F2 plant, that plant was short. Every time one or more gametes carried the T, capital T allele, and paired together, they produced a tall plant. The F2 generation had new combinations of alleles. Double tall, tall short, and short short.